Okay, so let's start from the beginning. So we have our cell here, and okay, we've got a cell here, and we've got our nucleus, we've got our chromosome, a couple of chromosomes in this nucleus. Okay, and we have a gene, and this gene that we're interested in is going to be turned on. So what do we call it when we say a gene has been turned on? What's a more correct term?
Express.
Gene express.
Right, gene expression. So we're talking about gene expression here. Okay, so we got gene expression. So what's the first step in gene expression? Oh, louder? Yes, transcription, right? So we're gonna get transcription. All right, so I'm gonna erase this chromosome. So we're gonna get transcription. Okay, so now we have an RNA. Let me see how this looks. Okay, now we have an RNA. And what needs to happen next? So what's this RNA? Describe it. So this is pre-mRNA, right? So pre-mRNA. Okay, so what's pre-mRNA? What are my boxes and lines?
Exon.
Introns and exons.
Exactly. A couple of people who answered. So boxes are exons. And these are the introns. Okay, what's the difference between the two?
The introns are the non-coding parts.
Okay, so the exons are? The introns are non-coding, the exons are?
Coding.
Right, the exons code for the protein, right? So what has to happen to this transcript? Oh, what's the next step? Splicing, and that does what? Right, it removes the introns. So now all we have left are the exons. Right, so we've gotten splicing, and now we just have that. So backing up, transcription, what enzyme is required for transcription?
RNA polymerase.
Exactly, and for splicing? I believe somebody, I think you said it, your audio is really, really weak. Spliceosome? Right, the spliceosome, okay? And it does the splicing, and now this can exit the nucleus. So we've got the transcript in the nucleus now, so it's now our mRNA, or mature mRNA. Now what happens?
Translation.
Right, so now we get translation. So for translation, what needs to happen?
The mRNA needs to enter.
Yeah, enter. Nope. Right, it goes into the ribosome, and with the help of what molecule?
Ribosome.
Yeah.
tRNA.
Right, the tRNA. We're going to make a protein, right? So we're going to get protein synthesis. Right. So here's our ribosome with the help of the tRNAs coming in. And what is the job of the tRNA?
It brings the amino acids to the ribosome.
Right, and how, well, do you know what order to put them in?
It matches up with the codons.
Right.
And use the according, the matching amino acid.
Exactly. So remember, the tRNA has the amino acid at one part and the anticodon below, right? So for AUG, you would have UAC, right? So that anticodon would base pair with the codon and bring in the proper amino acid. Okay, so that's how it's happening in our cells. Now, if this gene is turned on in every cell, what kind of promoter would it have? Maybe it's hexokinase. Right? Hexokinase, I think, is expressed in every cell in our body. What kind of promoter would you predict that hexokinase has?
Constitutive?
Right, constitutive or always on. Exactly. Or what if it's only expressed in one cell type?
Conditional.
Right, it would be a conditional promoter, right? And whatever condition is required would turn on that gene. Or in some cases, turn it off, right? Maybe it's on and it needs to be turned off for some reason. Okay, a condition is met and is turned off. Right, exactly. So that's the basics of gene expression in a eukaryote. Now, so now let's go to coronavirus. And specifically, of course, COVID-19. Okay, so what type of genome do we have? What type of genome do the eukaryotes have?
Genome?
Genomic, but what type of nucleic acid?
AGC?
Right, so DNA, exactly. So our genome is DNA. Is it single-stranded or double-stranded?
Double.
Right, it's double-stranded, right? It's a double-stranded. Each chromosome has 2 strands. They're double-stranded, and it's DNA. So we have a double-stranded DNA genome. Okay, the coronavirus does not. The coronavirus is a single-stranded RNA virus. Okay, so in the capsid, right, so we got the viral capsid, which looks nothing like this, but we'll pretend like it. It has a single-stranded RNA genome. Okay. So when it affects this cell, it kind of skips this part. Okay. So let's draw a new cell. Let's see how I can do this. So this is eukaryote. Now we're going to talk about the coronavirus. But we got to start with a eukaryotic cell. Okay, with our own genome. So we'll have our own DNA genome. Okay, and there will be 23 pairs of chromosomes in that nucleus. And now we've got the coronavirus. So the coronavirus comes along and it has, and I don't know how many copies of the RNA it has, but it probably has several in the capsid. So this circular thing would be the capsid, which is the protein coat that surrounds and protects the RNA genome. And then it has these proteins that stick out, and this is spike. Our infamous spike protein. Okay? And it just so happens, coincidentally, that that spike protein can bind to a receptor on the eukaryotic cell. Okay, so it can bind there, and when that does, the virus is invited to become part of the cell. Okay, so it's hijacking this ability to bind to this receptor, and that receptor says, okay, you can bond to this membrane and release your RNA. Okay, so now the RNA is in the cytoplasm. So there's an RNA in the cytoplasm, and one of the genes here is an RNA-dependent RNA polymerase. Okay, so it's RNA-dependent RNA polymerase. So what do you think that does? What does an RNA-dependent RNA polymerase do? Anybody want to guess? Or does anybody know?
Is it extracting RNA to DNA?
Not quite. Okay. So it's RNA dependent. So what does that mean? Well, first, let's back up. We all know, what do RNA polymerases make?
They replicate the RNA.
Okay, so this whole thing replicates the RNA, but specifically RNA polymerase. What does it do? We just talked about it back here, right? What molecules does it make? It says it. Its name. Right. Okay. In the chat. mRNA. Yes. RNA polymerase makes RNA. And in the case here, it uses DNA as its template. So this is a DNA-dependent RNA polymerase. So here we have an RNA-dependent one. So what do you think this template is for this RNA polymerase? What is it? It broke up.
RNA.
Yes, RNA, right? So it has the enzyme to make copies of this RNA. Okay, so it brings in the enzyme that will copy its genome. Okay, and that is replication. Okay, because we're making the same molecule. Just like when we replicate DNA, we make DNA. When we replicate RNA, we make more RNA. Okay, because they're started with an RNA template. If we start with DNA to RNA, it's transcription. So we have this molecule, this genome in the cell. It's going to use this cell's ribosomes, tRNAs, and all the nutrients to make this enzyme to make more copies of the genome. Okay, so it's going to actually use, sorry, what did I just say? Right, it's going to use RNA, TNA, and ribosomes to translate that gene so that it can then make more copies of the genome. Okay, so then we get lots of copies of the genome. It also uses the RNA polymerase, the cell's ribosomes and tRNAs to make other proteins, and you'll see those later on when I show you the genome browser. But there's about a dozen proteins, including spike and all the other parts of the capsid and some other enzymes that are translated in the cytoplasm to make more virus. So ultimately, you make more virus components, and these genomes get encapsulated with more virus, with spike proteins and other proteins. Right, and eventually the cell bursts and you get new virus out and to infect more cells in the body, or if you cough, this person coughs on somebody else, those viruses go to another person and infect another person. Okay, any questions about this? So we go from here to replication to new virus. And then the cycle repeats. Okay? So basically what the virus is doing is shortcutting this part. We don't need to do transcription. we're just going to make more copies of our RNA genome and translate that, and then have make more virus particles. Questions about that?
Why does it skip transcription?
Oh, because there's never DNA. This doesn't, as far as I understand, the RNA genome is copied directly back into RNA. Okay, there's no cDNA here. That's a different virus, which is also a single-stranded RNA virus, but it's a retrovirus. We talked about it early in the class, right? And that one has a very different lifestyle. So now let's talk about the Pfizer and Moderna vaccines. So this is the natural way that things happen. And now we'll go another way. I'm going to make my nucleus smaller so I can use the same cell. There. So the vaccines. And this is just the Pfizer and Moderna. Okay, both of them are RNAs. Okay, they're RNAs in a lipid droplet. And that droplet protects the RNA, but also allows it to go into the membrane, right? So you have a lipid droplet with an mRNA to spike. Okay, so we've got spike protein here, or the mRNA to spike. Okay, because spike is what is going to be eliciting the antibody response, the immune response. So they put in the mRNA for spike. Okay, and we're not generating and we are not looking for antibodies to the RNA. We need antibodies to the protein. Okay, so this lipid droplet fuses with the membrane. Okay, and spike RNA goes into the cell. So how do we get spike protein? This is RNA in the shot that they stick in your muscle, but we need the protein. So what's happening? How can we get a protein from our mRNA?
You have to translate it.
Exactly. So we're basically doing like the virus and asking the cell to translate this mRNA for us. Right? And so we do that. We get translation. And we get spike protein. And that gets put on the surface of the cell. Okay, and now we can go fishing for antibodies. So that's how it works. So we just are injecting the mRNA into the spike, into the muscle. That lipid lets it bind to the lipid membrane. The RNA goes in, and then we use the translation machinery of the cell to make spike. And we can make a lot of spike, a lot more than could potentially go into a more traditional vaccine. The difference between the two, any questions about this before I move on?
So the vaccine, we're actually making antibodies for the spike and not the actual RNA inside the virus?
No, we're not making the antibody. We're making the actual viral spike protein.
Oh.
Right, the body's gonna make the antibody. We'll come back to that. But this is the viral mRNA. We've just taken the one gene out, and we'll see this later in class. We've taken the one gene out. You can make these RNAs really simply, or fairly simply, in great big vats of bacteria, clean it up, encapsulate it, and then inject it so that it goes into the cell, and we translate it, and we get the actual spike protein. That's the only thing, so we don't get sick, right? It's not the whole virus. It's simply one mRNA. Good question. Any other questions?
Yeah, so that spike, is that the same spike that you're pointing to in the, yeah, right there?
Same one. So this is just from the natural virus. We've made this one through isolating the RNA, but the protein is the same.
RNA.
So when
So with the
sorry, go ahead.
sorry, you can go. So with the spike protein going on the surface of the cell, that makes it so the coronavirus can't bind and infuse with this.
No, we'll talk about that in a minute. We're not going to get into a lot of detail of immunology, but as cells do their job, do their functions, they routinely put proteins on their surface for the immune system to check. Okay, and if it's a normal cell protein, the immune system ignores it. But if it's a foreign protein like this spike, if there's an antibody that recognizes spike, then that anti-spike antibody will trigger the immune system. Okay, but this is a random event, and we'll come back to that. Okay, somebody else had a question.
No, she asked the same question I asked.
Oh, good. Okay, right. So cells just routinely put different proteins on their surfaces, even though that's not where they really belong, for the immune system to be able to check, is this cell making only what it should be doing, or has it been invaded? Okay, it's a pretty complicated system, so we won't get into it. But definitely, if you get a chance, take some immunology. It's quite fascinating. Right, so this antibody. We're not going to talk too much about it, but just so you kind of understand how the immunity works. These antibodies are generated randomly in our cells, okay? In the cells of the bone marrow that make antibodies, and the B cells, they randomly rearrange the chromosome so that they make unique antibodies. Okay, and there are millions of these things in our bodies. Okay, just floating around in the blood and in the lymph, and they're randomly generated. Now you may think, well, if you're randomly generating antibodies, you might make an antibody against self, and that happens. And those get screened out during the whole process of antibody development. So if it recognizes self, it gets killed off. That cell will get killed off, and others get to live and have the antibodies produced. And so randomly, people will have this antibody. And if this presented with its antigen, okay, so the spike protein is the antigen, then it triggers the immune system to go into full function. Okay. And then by going into full function, it goes into makes memory cells so that this particular antibody will stay around longer. You get a booster, so the immune system sees it twice and becomes really, trained that this is a foreign protein, and we need to basically tell this cell to die. It'll trigger the cell to either be triggered to do what's called programmed cell death, or it'll get engulfed by another cell and destroyed. Okay. But that's how we elicit immune responses with these types of vaccines. Now, like the AstraZeneca and the Johnson & Johnson, they're the more traditional vaccine where it's either dead virus or just a protein. So they're injecting the protein directly. And that's the way it's always been done. This is the first time the mRNA virus vaccines have ever been used. And as you see, they came out more quickly. These are faster things to develop. It takes longer to develop the more traditional type, and they also are going to be more responsive, and we're going to talk about that in a minute. Okay, so any questions about this? So this, we're not going to do much on this because the immunology is quite complex. But just recognize that you have random antibodies floating around in the blood and lymph, constantly checking cells for foreign to be able to bind. If they bind, they elicit a response. They activate the immune system, and the immune system takes care of the invaded cells. Okay?
Where did the mRNA spike come from?
They made it in bacteria.
And they put it into the cell.
So they make it in bacteria, they clean it, then they put it into these liquid droplets, these lipid droplets. Okay, lipids like fat. Okay, and then that goes in the vial and gets dried down. Then at the vaccine place, they'd add saline to the vial, resuspend these droplets, and inject that into your arm. And then because it's lipid, just like your plasma membrane is made of lipids, this is able to bind to the plasma membrane and release the RNA into the cell.
Oh, I see. Thank you.
Sure. Any other questions? So basically, we're taking over the natural processes of translation to make the protein that we want to get an antibody against. Just like the virus takes over to make more virus. But hopefully by getting people immune, we will prevent this because they will recognize the virus before it even gets to bind to a cell. Or if it does, it will limit your sickness because there's a lot of the antibody around already. So you may still be possible to get sick with being vaccinated, but it'll be a much shorter sickness and hopefully not as severe.
And one more question. The antigen was the protein?
Right. Antigen is the spike protein in this case because an antigen is what an antibody binds to. Okay. But spike protein also, of course, has a different function in the virus itself. Here we're saying, hey, this is what we want an antibody against. Like, let's go find it. And it really is a fishing. I mean, it's random. And so, but because so many antibodies are made, it's possible that it's already in your body.
So like if antibodies are like always random, say like the vaccine never happened, how long would it take for like a antibody to form for COVID, like on its own?
Right. So it's similar to so basically, right, with the antibody, what we're doing is we're activating the immune system without getting sick. Right? You might get without getting sick. So you don't actually get COVID disease using the vaccination. Okay? People who react to the vaccine and feel icky the next day, that's the immune system making them feel bad, not the vaccine itself. Okay? The immune system, when it's activated, releases interferon, and interferon makes you feel like you have the flu. Oh, and the question is, why the second dose worse than the first? Anybody want to hazard a guess? Why would the second dose be worse? So with one dose, right, this has been going on, I don't know how long this goes on in the body before this disappears, but it's probably several days, if not longer. So with the first dose, right, it's taken time for this antibody to find this antigen, and get the immune system kind of going. So what about the second time? Oh, and it's the same dose. Good question. Right, both doses are the same. For Pfizer, I believe it's a half a milliliter, and Moderna is I think a full milliliter. So that's a good question. That could be maybe the second dose is bigger than the first, but they're not. They're equal. And somebody said, right. So basically, the body is primed from the first dose of the vaccine, and so it elicits a stronger response the second time. Right. So first dose immunity is about 65% of the people are immune. 65%, right. Second dose, you have like 95% effectiveness. And it's because you're eliciting this stronger and stronger response. And these are some of the most effective vaccines ever developed. The flu vaccine, I think, is 65%, right? And it's not a really fair comparison, but it is. That's about the flu vaccine. Other vaccines, not quite as good, but 95% is pretty amazing. Right, so basically, somebody said they knew somebody who had COVID disease, then they got the, and then you're supposed to wait a month, I believe, or something like that, and then get vaccinated, and then they got really sick, symptom-wise, but not obviously not disease-wise. Right, they already had an immune response, and so the booster really, pushed the immune system one more time. Right, so the question is because the immune system's working harder, it's not necessarily working, the response is larger, right? The response is a bigger response. Because this antibody, one thing that happens during the initial immune response is the cells that make that antibody multiply so that there's now more. The body now is aware that this is an intruder, and it makes this big response so that there's a lot of antibody floating around in your body at this point. So any new response is going to be pretty strong. And then, right. But then over time, that fades. Right. So after about 10 years, if your body hasn't been presented with this antigen, the immunity goes down, way down. Right. So like by the time you're in your 30s, the MMR you got, the Tdap you got, all of that, you won't have immunity to those. So they are. It's a good idea for adults to get boosters for all of those. And if you've been to the ER lately for a cut or a scrape, one of the first things they do is stick you with a tetanus shot. Right? You don't actually get much choice in the matter. And they'll do a tetanus within 5 years, if necessary. Because the immunity does fade over time because it's not being challenged. Any questions? These are good questions. Anything else? Great. Okay. So then, like I said, the difference with Johnson & Johnson and AstraZeneca is that those are proteins or weakened virus. I don't know which. I haven't looked into how those are, but that's the traditional way. You either take specific antigens from the virus and put it in the shot, or you take the whole virus, weaken it in a way that it can't make a person sick, and inject that.
I have a question.
Yeah.
Is there a reason why Johnson & Johnson is one dose, whereas the other two are 2 doses?
I don't know. Okay, biologically, no, I don't know the reason. And those are determined by trials, right? So that's why. So it's basically determined by trial that we needed 2 doses to really push immunity for these. And it could be because they're brand new, they decided to go with a 2-dose regime just to make sure it worked. That's another potential. It's like, let's hedge our bets and do 2. Many vaccines are 2 doses, you just don't remember it because you got them when you were an infant. But most of the vaccines that I'm aware of, except for like flu, are multiple doses, at least 2.
Dr. Burnett,
Yep.
do you know why some of the Johnson & Johnson vaccines produce blood clots?
I don't know the biology, no. I don't know if it's understood either. And like people keep saying it's like 6 people in millions. And it's in the same group of people. If the same number of people got coronavirus, 200 would get blood clots. So, but why it's triggering it, I don't know. And I don't know if, we could look later and see if anybody knows. But basically, the only people in Europe and in the United States that got blood clots were females of a certain age range. So if somebody falls in that age range, just get this one, right? One of these 2. If you're a guy and you got Johnson & Johnson, probably not going to have an issue. Don't stress it over it. The nice thing about Johnson & Johnson being 1 shot is that it's really good for people that are hard to get to, right? So people who are shut in at home and can't get to clinics, people who live in really remote towns where they may not have a lot of access to healthcare, right? 1 dose and they're done, which is good. It just so happened that these were introduced first, and so that's why most of us are getting those 2. How are the dead vaccines collected? I'm not entirely sure. I imagine one way is to treat them with ultraviolet light or other radiation, which would really mess up their genomes and prevent replication. An article about, I haven't heard anything about it affecting women's menstrual cycle. That's interesting. We'll have to look into it. We share the article in the chat. And there's going to be, this is vaccinations of a scale that we haven't seen in a long time. So very rare side effects are going to come about, but we don't need to, I don't think people should stress out about them. Any other questions?
So like you said in the Johnson & Johnson vaccine, they use like a weakened version of the virus. So like, even though it's weakened, could the person getting it still spread the virus?
That's been questioned and I don't know the answer. It's probably highly unlikely because it's weakened so that it can't do the replication. Basically, it's circulating in the blood so that antibodies are found in the blood. Right? The whole idea is to prevent people from getting sick with the vaccine. And the smallpox vaccine was an actual live living virus. They scratched the skin, put the virus on, or it's actually a weakened virus, but it was just enough to make 1 pox. And that's how you got immune to it. But then, but you guys don't get smallpox vaccines. And then you might read, if you read up on this, you might read that there was a bad batch of polio vaccine, and that did result in people getting the vaccination, but that was a bad batch of vaccine. It didn't go through quality control. I don't know how it happened, but there is vaccine-derived polio circulating in certain areas of the world. For the most part, polio is almost gone. Okay, let's see. I'm going to talk about a couple more things and then we'll take a break. So what is going on then with these variants? What do you think the variants of the virus are? Or what are they if you've read or heard about it in the news? What are the variants?
Is it the like virus changing shapes so the antibodies that we have formed won't like recognize it?
Okay, that's one. We haven't seen that yet, and that's called a breakthrough virus. Okay, but how does that happen? How does the protein get modified? Where's the variation actually occurring? Right, so Enrique says the virus has a different gene sequence. Exactly. So how does that happen? How do we get a different gene sequence during this replication process?
Maybe possible mutations?
Okay, and from where? What's the source of the mutation? Not other DNA. It's the polymerases, right? The polymerases make mistakes. And they're random. Again, here's this randomness, right? They're random mistakes. Some will make the virus so that it can't function, and those variants will go away. But some will make the variants better at infecting cells, and thus will outcompete the original virus and become dominant. Okay, that's called evolution, right? And so that's what we're seeing. Especially the real worry, not only for the health of the individual, but the long haul COVID patients, these patients who are 3-4 months in the hospital, the virus is replicating billions of times, right? So it's had a chance to probably mutate every nucleic acid, every base of this RNA, and eventually most of them will probably make the virus dead, but some could make it a better infector, and so outcompete the original one, and could result in just the right change that this interaction won't happen anymore. And that's called a breakthrough virus. Okay, so that's it. That's what we were talking about, non-synonymous and synonymous changes, right? It's RNA polymerase in this case, and for humans and other people, other organisms with DNA, it's the DNA polymerases. Okay, they're error prone. And that's the basis of evolution. Most of the variation is actually during replication. And the genome here is DNA, or the replication of the RNA genome here. Now the interesting thing, this is the first time this is kind of, I think, ever happened. So we have 4 vaccines right now to the virus, right? So let's say it escapes the Pfizer, somebody has, right, the virus develops a way that it can now infect people that got the Pfizer but not Moderna. So if a Pfizer person comes in contact with this person, it'll stop transmission of the virus. Right? Or if somebody got Johnson & Johnson and it escapes that one, these will stop the virus potentially because they have listed different antibodies. Right? So having multiple vaccines is actually going to help prevent these breakthroughs if we get people vaccinated fast enough. Right? That's the big problem. Right, so that's the variant. So there you go, it's polymorphism. Right? So now we've discussed everything in the first 4 weeks except for bacterial transformation, but all the biology is the same, right? It just depending on where you start. Here we started with genomic DNA, here the genome was RNA. Okay, so that's it. Right, nice. Third the motion, get vaccinated. And again, if the Johnson & Johnson makes you nervous, just sign up for one of these 2. This is the most common anyway, so you'll most likely get these 2 vaccines. Right? And if it's not available in your area, you can go somewhere else. They don't really care anymore, they just want people getting vaccinated. I want to leave it with this, and then we're going to talk about the RNA test. So the Pfizer RNA is just a plain old RNA, just like we would make in a cell. You purify it. This is made in a bacterial cell. It's purified, put into the droplets, and then used for the vaccine. The Moderna is a modified modified RNA. And I haven't looked up what the modification is. So Pfizer is just plain old mRNA. And so it has to be stored at minus 80 degrees Celsius. This is really cold. Okay. If you think about it, I'll take a picture of one of our minus 80s that we have here in the building, and so you can see what a super cold freezer looks like. The Moderna is a modified RNA. And so it's okay at 20 degrees Celsius for long-term storage and can survive high temperature, meaning room temperature, for longer periods of time. So why is this? What's going on? So I'm going to draw just very simple chemistry of an RNA. So we have an RNA strand here. So here's the ribose sugar of RNA. Oh, that's not going to be visible on the camera. Hold on. Here's our RNA sugar. And you don't have to worry about remembering this. You'll learn it later. Phosphate, it keeps going. Here we have another one. And this may not be accurate because it's been a long time since I really studied these molecules. Okay, we'll be able to see that. Okay, so and then off of here would be a base. So maybe this is an A. And off of here would be a C. Okay, so this is the backbone of RNA. Oops. This is DNA because that is an H. And if you haven't had enough chemistry, the angles are carbons. So there's a carbon there, 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 and there. And up there, I didn't draw it. And then if there's nothing here, is an H. Okay, but I'll put it in. So that would be DNA. So that's the deoxy part of DNA. RNA has an OH. So it has this hydroxyl group. And this is the problem for scientists. Obviously it works just fine in the body. And that's why there's this difference. This oxygen in water or in solution will attack that phosphate and you'll lose that bond. So now you've got this circular thing here with the ribose and now that, and we've lost that bond. So we broke the chain, right? So at room temperature, RNA will degrade just because of that, attacking that, okay? That's what makes it hard to work with RNA, one of the reasons. I mean, obviously you keep everything on ice, but it makes it challenging. So that's why this one has to be stored super, super cold. You want to prevent degradation of the RNA just by it attacking itself. They modified the bases of the Moderna one, and I don't know what the modification is, but it prevents this type of degradation. Okay? And that's why it can be stored at minus 20, which is your freezer in your house, roughly. And here's the problem, and this is where the misinformation and people exploiting fear and misinformation. Okay, this is a modified RNA. Now I've heard eavesdropping on people having conversations that this RNAs are modifying. Okay, so the RNA, so instead of a modified RNA, they are calling it a modifying RNA. What's the difference between those two meanings?
Modifying means it's causing change, and modified means that it is like change itself.
Exactly. Right? But see how insidious you can be just by changing one thing and scaring people. The conversation I overheard was, and I've read this in the paper as well, is that this modifying RNA goes into the nucleus and modifies our DNA. Right. So, and you're exactly right. Here, the RNA itself is modified, right? Like you modify a car, you put a lift kit on it, right? You've modified that car. Modifying would be that car ran into something else and busted it up, right? It modified another car, right? So it is the agent of change, which is completely wrong. Okay. The only thing that happens with this RNA is it gets translated. So there are people out there, we won't get into the reasons, that don't agree with the vaccination process, and so they look for ways to really mess with people's understandings. So if you hear people talking about this and you feel like correcting them, you can try. Or at least make sure you use the words correctly when communicating with other people. So again, precision in biology is so important, right? Any questions about that? And I said we would stop now, but not. One more thing. The Johnson & Johnson and AstraZeneca will not be able to keep up with the variants as fast as these. Why do you think that is? So as we get variants, and they're sequencing them, not fast enough, but they're being sequenced. Pfizer and Moderna can react much more quickly than Johnson & Johnson. Why do you think that is?
Though? Is it because the Pfizer and Moderna, they're making the spike protein instead of
Right. But we're seeing changes in spike, so there are variants that have differences in the spike. So how can these guys be quicker to respond than Johnson & Johnson?
Would it have something to do with it being 2 doses instead of 1, like the Johnson?
Okay, possibly, but would the second dose be identical to the first dose?
Yes.
Well, what if you know that there's a change, there's a SNP, right? So we have some variants out there that have an A, and we have some variants out there that have a G, and we know that variant with a G is more aggressive. But what's in here has the A. How could we modify the vaccine? Right. If we know the sequence, couldn't we just make a new RNA and stick it in the vaccine? And that's what they're doing. It's already in trials. So these variants, they're already modifying the vaccine so that it will not only have the original strain that started last year or so, but now these new variants are also being included in the and I think I read that Pfizer has gone into phase 2 trials with those. So basically what I expect is that perhaps even before we all come to campus in the fall, we might need a third booster, right? And that's because of the variants. And probably every year we're going to get a booster for coronavirus, just like we do for flu, right?
So I had a question.
Yep.
They're putting the mRNA with those variants into the vaccine already?
In trials. The ones that are being used to vaccinate people, no. Like what's out there now, not yet, because it has to go through the trials. But they know they're safe already, they just gotta show that they're effective. So the trials will be really quick. So let's talk about the RNA tests. Okay, how do we detect COVID in human tissues, basically saliva? So let's remind ourselves, what is the genome of COVID-19?
RNA.
RNA, exactly. So it's an RNA virus, so there should be, if this person is infected, they should have coronavirus RNA detectable, right? So how would you do that? Let's design the experiment. Okay, so you go to a collection site. The one I went to is you swab your cheeks. Other people, they shove a stick up your nose and swab your sinuses for you. I only did the cheek swabs, and then you put it in a tube that looks a lot like this, right? It has a little bit of liquid in it that protects the RNA, because remember we just said RNA degrades pretty easily. So we put it in a tube with a chemical that protects the RNA, and where I went, you threw it in a great big trash can, and later that night they locked it in and ran the tests. Okay, so we collected our sample. Now, what do you think they do at the facility? So we collect the sample. So what would be the next step?
Run PCR with primers for that.
Okay, so you jump 2 steps. So let's back up. You're on the right track. So we are going to do PCR and put primers to COVID. Exactly. Nice. But we jumped over 2 steps. So we got a cotton swab full of spit, right? Or snot, depending on where it came from. Mucus, right? So what are we going to do?
Extract RNA?
Exactly, we're going to extract RNA. Now, when we extract this RNA, are we going to have just virus RNA? Do you think we're going to extract only virus RNA, or will there be other RNA in there? Other, okay, where'd that come from?
From us?
Yeah, human. So it'll be viral and human if the virus is present. And there better be human RNA in that, right? If not, something went badly wrong. Because when you swab these tissues, whether it's your nasal cavity or your cheeks, inside your cheeks and your gums, you're pulling off cells. And if we were in class, we would have, during microscopy, I would have given you swabs, and you could get some saline and make a slide and look at your cheek cells. Okay, they just come off really easily. They're great big round cells of huge nuclei. They're really easy to see with a microscope. So yeah, we're collecting cells, and so when we extract RNA, we're going to definitely get human and maybe viral, right? Because that's our question, right? So then we've extracted the RNA, now what do we have to do? Can we do PCR right directly?
No, because we need a double strand.
And it needs to be what form?
Genomic DNA?
Not genomic. You guys know this, we've been talking about it since the first day. cDNA, right? We gotta get cDNA. How do we do that?
Reverse transcription.
Exactly. So we've got our RNA, mRNA or whatever, it doesn't matter, all the RNA, and we do reverse transcription. And what's the enzyme?
Reverse transcriptase.
Exactly. And now we have our cDNA. Now we can do PCR. Right? Because there's a limitation of PCR that we have to have cDNA or DNA. The C or G doesn't matter. We just need DNA form. So this copies mRNA to cDNA. Now we can do PCR. And we'll throw in here design primers, because we'll do that next. Because what are the primers for? Why do we need the primers? Okay, and what is it? You're right, but what is it? Not the cDNA. What does the
Like is it the whole cell? Like it knows.
No, you're on the right track. So the PCR, the primers direct something to copy a specific region. What is that something? What's in the PCR?
Right?
Not RNAs.
Is it the
What's the enzyme?
polymerase?
Right, the polymerase, right? So it tells the polymerase where to replicate the DNA, right? We call it TAC, right? But TAC is the DNA polymerase. TAC is the brand name, but it is the enzyme. So we copy this, and then what can we do? What's next? We've done reverse transcription. Now we've got our PCR with primers. What's the next step?
RNA, run a gel.
Run a gel. Yep, gels. And that's basically it. We could sequence if we want to, but we don't need to for this test. Okay. Yes, those are the steps for designing this experiment. Now let's think it through. Let's predict. If somebody is infected with COVID-19, what's the result? What would we see on this gel if the person is infected? What would we see?
We have to perform a positive control, and if we see a result under the positive control,
Okay, excellent. You're getting ahead of me. You're anticipating my question. But let's think about it. What would be somebody with a positive result? What would we see? If we had our COVID-specific primers and we run our gel, would we see a band or not in a positive patient? And this is different from a control, right? If a person's infected, yes, we would see a band, right? If the person's infected, we'll extract the RNA, make the cDNA, and then amplify that RNA. So we should see a band on the gel. What if the person's not infected? Would we see a band? No, right? No band. No result. Now you guys haven't done PCR for real, but let me tell you, it's fairly easy to get no band in PCR. There's many, many ways. You forget a primer, won't work. You forget to add the right amount of water, it won't work, right? You don't add the sample template, it won't work, right? Remember our RNA, how easy I said it is to degrade? If it degrades, no band. So is this conclusive result? Can I say, if I just see this result, no band, can I say the person is negative?
No, because it could be a false negative.
Exactly. You could be a false negative because of all those reasons and more. So what would be really, really useful? What's something that will always give us result regardless of viral condition? What could we include? And this is getting back to the positive control that was mentioned. What could we include in our experiment that would always give us a result if everything was done correctly?
Have the virus itself.
Okay, so maybe we spike it. We could try that and we'll come back to that. Look, there's something else that's already in the sample. It's on the board.
The human RNA?
Do what?
Is it the human DNA?
Right, well, it's RNA, right? We've extracted human RNA along with potentially viral, so there better be human RNAs in that sample. So if we pick a human gene, it better amplify. And we're gonna choose the gene P30. You better get a band. Now, if we get this result, no band for COVID, and a band for human, we're pretty confident that's a true negative. Right? Does that make sense to everybody? So this is our positive control. This tells us that we extracted the RNA correctly, we made our cDNA correctly, and we made our master mix correctly. Okay?
That result. So if a band appears under the human gene, that means the
That means this, we can trust this negative result.
negative is true.
It's most likely. There's still potential for there to be a negative, a false negative, but most likely it's a true negative. Okay, there's always room for false negatives for various reasons, but for the most part, we could believe it. It might trigger a second round of testing just to make sure, retest the sample, repeat it, but you probably would just call this a negative. So that's what we're going to do. We're going to design primers to two genes, two COVID genes, the human gene and the spike gene. And the spike gene is something that we add to the mix, or we use as another positive control. Okay, so let's talk about primer design now. Any questions?
Okay, thank you. Yes, I have a question. So what is happening when people get an inconclusive result back?
Well, assuming that they've done all of their controls for the test. Wait, did you say inconclusive?
Yes.
Okay. So it could be, well, first off, they would always do their controls, and the way this is done, it's done in a huge batch. So if all the positive controls that they use are good, an inconclusive result, the CDC requires 2 COVID RNAs to be tested. Okay, and that's what we're going to do now. And both need to be positive, at least in what we're going to call a positive test. Both bands need to amplify. If one does and one doesn't, that's inconclusive. Right? Exactly. Good question. So you're going to design an experiment, and it's modified. This isn't exactly what the CDC's test is, or the tests that are being used, but it's very similar. You're going to pick 2 COVID RNAs, and you'll see in a minute there's like 16 different genes. I've given you the sequence to human P30, and the CDC or somebody gives us spike RNA. Okay, so we're going to design the primers to these 4 things, and it doesn't take long. We'll be able to finish this up in like 10 minutes. Right, and then you would set up, in this class, we're going to do it where you would set up 1, 2, 3, 4 different PCRs and do the test. Okay. It's not the best way to do it, but I don't want to complicate things any further.